Well, the first thing I'd like to say is that everything I'm going to talk to you about today is the product of a collaboration with my wife, Maureen Neitz, shown here. Well, as you already heard, I did my PhD at the University of California in Santa Barbara. And at the time, it was somewhat of a golden age for neuroscience. In 1981, Huebel and Wiesel had just won the Nobel Prize in physiology of medicine, and medicine. And I think there was great enthusiasm for this whole kind of approach for single unit recording that at different localizations of the visual systems that people were going to be able to figure out exactly how the brain worked. And there was very much enthusiasm for, the idea that ultimately this is one of the goals of neuroscience, is going to figure out how the brain works. And I did my PhD with Jerry Jacobs, and Jerry, he was a student of Russ Develoy. And Russ Develoy was the very first person, he was a pioneer, and very first person to ever record from deep structures in the primate brain. And he discovered opponent neurons in the lateral nucleus of the thalamus. And so he's the very first person to really discover the basis for color opponency in the primate. What this means is that with Russ as my grandfather and Jerry as my academic father, then I'm a third generation color vision scientist. Also at the University of California, just right before I arrived there, David Primack had been there, and he's a pioneer in the study of chimpanzee language. And Jerry told me that David told him that the goal of neuroscience is that we needed to crack the brain code. I'd never heard this expression before, but as a graduate student, I was very excited about the idea that ultimately that people studying the nervous system would be able to, quote unquote, crack the brain code. Well, there was no shortage of great ideas about how the brain works. But the problem is that there's all these different ideas, but we have to figure out which ideas are right. And today I'd like to talk to you about our recent attempts to test some long-standing ideas about the workings of the visual system using color vision as a model. So just thinking about the way that people think about the brain, prior to the 20th century, it was often conceived that information from our senses was conveyed to a quote unquote sensorium, which was the seat of sensation, where impressions from the external world are conveyed and perceived. So it's very much an idea that that the information, the whole problem of understanding the nervous system was understanding how ultimately information was coded at the level of the retinas and ultimately conveyed to some central location that was where it was interpreted as our conscious vision. And just for an example, I'm showing you a quote that appeared in British and Foreign Medical Review in 1845. And here's the quote. "What we see is not the image formed on either of our two retinas, but the mind receiving both blends them by an unconscious act into a representation of the object." So the idea is that the whole problem is that the information is available at the eyes. It somehow gets sent to some location in the brain that's responsible for vision. And with this in mind, it's, at least from this perspective, that many ideas, even to this day, have focused on trying to understand the nature of the information transfer. Basically, how is the information coded at the level of retina, and then really, how does it get to some central location where ultimately perception happens? So here's a first idea about the way our nervous system works. We have this very strong intuitive sense that we have conscious access to all the information in the retinal image. That's idea number one. And it's now well understood that the human retina contains a diversity of ganglion cells, and a subset of those ganglion cells are shown in this diagram here. And so a second idea, again, very intuitive one, is that ganglion cells are primarily concerned with visual perception. So as an example, here's a quote from a paper published in PNAS that came from Marcus Meister's group. The quote says this: "The retina reports the visual scene to the brain through many channels." And so I would think most of us assume that when we talk about reporting a visual scene automatically makes you think that the ganglion cells are concerned with conscious vision. Otherwise, why would we talk about a visual scene? Okay, and a third idea that is still often brought up today is the idea of efficient coding. And so the efficient coding hypothesis was originally proposed by Horace Barlow in 1961 as a theoretical model of sensory coding in the brain. So the idea is we know that there's a lot of different parallel channels, and in order for the information to be efficiently coded, the information in the retina would be encoded across many parallel channels in a way that minimized the number of spikes needed to transmit the given signal. And efficient coding hypothesis grew out of information theory, which provides a mathematical framework for analyzing communication systems. And so in this case, efficient coding is talking about the case where there's communication between the eyes and the brain. Again, the idea is that ultimately all the information that's available in the images on the two eyes is basically broken down into a whole bunch of different communication lines that are all independent from each other, so that you can officially send all the information to, at least in one idea, someplace where it gets reassembled and gives us our perceptions. The thing that the efficient coding hypothesis predicts is that information carried by different ganglion cells should be uncorrelated. So, as we already heard today, color vision has been considered to be a model for testing ideas about how the brain works. Well, hue perception is concerned with computing the surface reflectance of a reflectance spectra of objects. So by knowing the surface reflectance of an object, you can instantly tell when meat is cooked. You can tell when fruit are ripe. Or you can tell when your friend is blushing from embarrassment. And the S, M, and L cones serve as detectors. And they're sensitive in 3 different spectral bands. So one spectral band is in the short wavelengths, one in the middle wavelengths, and one in the long wavelengths. So basically, if we think about our visual system as far as color is concerned, is a spectrum detector, you have only 3 different spectral bands, then there's 8 distinguishable patterns of reflectance across those 3 bands. For example, one pattern of reflectance could be that the reflectance across all 3 bands is about equally high. An object that we perceive as white has high and about equal reflectance in all 3 bands, the short, middle, and long wavelengths. Conversely, objects that we perceive as black have low and about equal reflectance across all 3 bands. So those are the 2 of the possible relative reflectances that you can have if you just have 3 different detectors. And then there's other possible classes of spectral distribution in where there's lower reflectance at one or more of the bands and higher at the other, so that now you have differential reflectance across the bands. And if you just have 3 different bands, then there's 8 possible ways that you could now have different reflectances. And we already talked about 3 of them. They could be all high or all low, and there are 6 others. So, shown here, you could have low reflectance in the short wavelengths and high in the middle to long wavelengths. Or conversely, short wavelengths could be high in reflectance and long wavelength could be low. You could have just high reflectance in the middle wavelengths and low reflectance in the short and long wavelengths, or the converse. And finally, you can have high reflectance in the short and middle wavelengths and low in the long wavelengths. So it turns out that there's, you could classify, the different possible spectral reflectances into those 8 different categories if you have 3 different detectors. Well, each classical color channel that was originally posed by Hurvich and Jameson in 1957 can be conceived of as a detector of one possible pattern of relative surface reflectance. So according to the Hurvich and Jameson model, there was a hue channel responsible for the sensations of green that adds together the long and short wavelength cones, and those are different from the middle wavelength cones. And so that green channel could be thought of as being a detector for reflectances where the reflection is high in the middle wavelengths, but low in the short and long wavelengths. The interesting thing is that there are 8 possible spectral patterns, but we only experience 6 basic color sensations. We experience black and white, the achromatic sensations, and then 4 different hue sensations: red and green, blue and yellow. So according to the original opponent process theory, as proposed by Hurvich and Jameson, that the color channels each received inputs from all 3 cones. So there's 3 different, according to the theory, there's 3 different color opponent channels: a blue-yellow channel, a green-red channel, and a white-black channel. And in their theory, that all the channels get input from all 3 different cones. So the blue-yellow channel takes the difference between the short wavelength cones and the sum of the middle and long wavelength cones. And according to their theory, the red-green channel took the difference between the middle wavelength cones and the sum of the short and long wavelength cones. And also, according to their idea, that black and white also, those channels also got input from all 3 different kinds of cones, but the 3 different kinds of cones are just summed together. So we can see that there's we can think about things in 3 different columns here. The first of all, that there are 3 different proposed circuits. There's one that compares short to middle and long wavelength cones, a second circuit that compares middle wavelength cones to long and short, according to this theory. And then there's a third circuit that adds together the 3 different kinds of cones. And that each one of those could be thought of as a detector for a particular spectral distribution of reflectance. And then ultimately, those different spectral distributions could correspond to different hue sensations. So the S versus LM cone process, for yellow and blue, the M versus L plus S for red and green, and then all 3 added together for black and white. The thing that's interesting is that, at least from the pattern of different reflectances, there's an extra set of possible reflectances in the world, but there's no corresponding sensation that goes with those. So that's the Hurvich and Jameson model. And the one thing that when we make a theory that basically idea that our color vision is based on some actual circuit in somewhere in our visual system that compares the cones in some ways, that's just a theory. But if we make that our hypothesis and say, okay, that we're going to assume that someplace in our visual system, there are actual circuitry that compares the different kinds of cones, and those circuits are responsible, the activity of those circuits are responsible for our hue sensations. If that's true, under that assumption, it turns out that the blue-yellow channel cannot be based on a circuit that compares short versus the sum of the long and middle wavelength cones. And it turns out that even though the primate visual system has circuitry, the small bistratified ganglion cells, that has S versus the sum of N plus L cone inputs. And it also has ganglion cells with L versus M inputs. And those have often been proposed to be the basis for our hue perception. If we assume that there are actual channels that have cone inputs that are responsible directly for our hue sensations, that the color opponent hue channels do not correspond to the cone inputs of either of these types of ganglion cells. And this is very well illustrated in this diagram that comes from Russ Develoy. And first of all, he plots out the spectral sensitivities of cone opponent neurons, and he's showing the excitation side in the cone opponent neurons that have short wavelengths as the short versus LM, middle versus long, and then the sum of L plus M versus S. Those are shown in the bottom panel. But we can see that actually, if we measure our perception, and this particular data was measured using hue scaling, we can see that our actual hue perception doesn't correspond to the spectral sensitivities of the cone opponent neurons, the most common ones that are measured at the level of the LGN. And the reason for this is because, for instance, the peak of our sensation of blue appears somewhere between the S and the M cones. And the reason for that is because if there is such a circuit, it has to add together the short and middle wavelength cones in order to account for our hue perception. And the other thing that we see is that there's a reemergence of a red sensation in the very short wavelengths. And this again would have to be because there's the short wavelength cones have a contribution to our sensation of redness. So we can also just look and see the same kind of thing if we just compare the spectral sensitivities of the three different kinds of cones to our hue perception across the spectrum. And we can see that, that the location of unique blue actually occurs at a place where there's actually a higher quantal catch of the M cones than the S cones. And again, that an explanation for this is because our sensations of blue are based on a circuit inside of our visual system, someplace, that adds together the short and middle wavelength cones, compares them to the longer wavelength cones. And the other thing we see is that the very short wavelengths, we have this very strong sensation of redness. Again, according to the theory, this is based on the fact that there is a circuit somewhere in our visual system that actually adds together the short and long wavelength cones, compares to the M cones, and in order to give us our red and green sensations. So this is the original opponent process theory from Hurvich and Jameson. And the difference between that theory and a modern opponent process theory is that we have to move the input from the M cones from add together the L in order to have blue-yellow coloration. We have to move it over so that actually we add together S and M cones. So that's the difference between the original idea of Hurvich and Jameson and what we need to do in order to account for the cone inputs to our hue information. And so to complete the theory, to give a modern updated opponent process theory, what we see is that according to this theory, there are three different kinds of channels. There's one that's exactly like that Hurvich and Jameson mentioned, that compares M versus L plus S for red-green. And there's one that adds together S and M cones in order to give us blue-yellow. And in this particular case, we see that there's a very big difference now between what patterns of spectral reflectance are detected by the different channels. So now we no longer have a hue channel that corresponds to spectral reflectances where that would really respond to S versus L plus M and be really a great detector of spectral reflectances where the short wavelengths are low and the middle and long wavelengths are high. So There's 8 possible classes of patterns of spectral distribution, differentiable with 3 kinds of cones. And you might expect that there would be 8 different hues responding to the different patterns, or 8 different colors, but we only see 6 different colors. And that we really don't have an actual, according to the theory anyway, we don't have a circuit anywhere in our visual system that would be a detector of patterns where short wavelengths are high relative to middle to long wavelengths. And we argue that if information was fully encoded, we would be able to differentiate 8 different patterns of cone activity by different sensations, but we only get 6. The fact that hue encoding fails to represent 2 of the 8 reflectance patterns means that some color combinations that produce very different patterns of cone activation are indistinguishable. And here's an example. So if we take a light that from the spectrum down near 420, it has a color sensation that looks violet. But it turns out that we can produce exactly the same color sensation by mixing together a color that appears unique blue, somewhere between 470 and 480, and a longer wavelength light that appears red. Those 2 things give a bluish red sensation that we call violet. And it's possible to arrange it so that even though these things produce very different cone activations, they produce very, very similar color sensations. So here's an experiment we did just to see, whether or not people actually can match a mixture of 470 nanometers, a blue color, and a red color to a violet 430 nanometer. And so in panel A here, we show the spectral sensitivities of 3 different kinds of cones. And the 430 nanometer light produces a very strong quantal catch in the S cones, but relatively low quantal catches in the fairly low quantal catches in the middle long wavelength cones. On the other hand, the mixture of 400 and 630 nanometer light produces high quantal catches, relatively high quantal catches in the middle and long wavelength cones, but relatively lower quantal catch in the S cones. But it turns out if you give somebody a 430 nanometer light and you ask them to adjust the mixture of 470 and 630, the people are able to produce a match. And here they are plotted for two different subjects, and they were able to exactly match the hue of these two different lights. As a matter of fact, they could arrange it in such a way where even though these things produce such a strong differential activation across the 3 different kinds of cones, they're indistinguishable. And the reason, according to the theory, the reason for this is that the sensation of blue is actually served by both the activity of the M cones and the activity of the S cones. And so that ultimately when you take a 470 nanometer light, much of the sensation of blueness is actually served by the M cones. And the sensation of redness is actually served by both S cones and L cones. So that when we only are stimulating the short wavelengths, that the sensation of blue and red are mostly based on the S cones. But if we go to the longer wavelengths, then sensation of blue is based on M cones, and the sensation of redness is partially based on the longer wavelength cones. So that would explain, according to the theory, of why it is that these things look exactly the same, because our hue channel is actually adding together S and M cones. So 2 lights produce 2 different patterns of activity across the 3 kinds of cones. And the difference between these patterns of activity is easily distinguishable by a spectrally opponent Ganglia cell that compares S versus LM cones, because these two stimuli produce very, very different activities in any kind of a circuit that would compare S versus L plus M cones. And of course, we know very well that our visual system has ganglion cells that are S minus LM opponent, the very, very well-known small bistratified cell. Even though we have these neurons in our visual system, there is no hue percept that's associated with these stimuli that strongly produce differential activation of S versus M plus L opponent neurons. So that's extremely surprising because this would produce very big activity, but it doesn't produce any kind of a sensation that differentiates the color of these two different patterns of light. But what's even more puzzling is that the small bistratified cells are not the only cells in the ganglion cells in the visual system that have S versus LM opponency. In order to study the ganglion cells that have S versus LM opponency, we have been using serial electron microscopy to study the circuits in the primate retina. And here's what we've been doing. First of all, we started with a block of primate retina, and it's placed inside of a scanning electron microscope. And inside of the same microscope, there's also an ultramicrotome with a diamond knife. And so the experiment starts off and scans the surface of this block of retina and gets an image of a slice at this particular level. And once that image is taken, then the diamond knife takes a very thin section off of it, some 50 to 90 nanometers thick. And then once that section is gone, imaged, again with the scanning electron microscope. Repeat this process of slicing and imaging thousands of times, and ultimately you get a volume of many slices of retina. And then we use a computer in order to assemble this 3D volume into something that we can go through and manually trace out and basically study all of the anatomy of all the circuits in the primate retina. Just to give you an idea of a little bit what it's like, here's a movie showing us moving progressively through different slices at a very small location. This is a location right underneath a single cone pedicle, and show how you can trace out a particular process and follow it through from section to section, and then use those tracing ultimately to reconstruct a circuit. In this particular case, what we reconstructed are the terminals of the midget bipolar cells, just as they're interacting with an L or M cone pedicle. Well, luckily, it turns out that the terminals of the short wavelength sensitive cones are distinguishable by their anatomy from the terminals of the middle to long wavelength cones. And so here's an image that was taken at a section that's right at the level of the terminals, the terminals of the cones. And so each one of these little outlines is just an outline of a cone terminal and shows this little mosaic. And we were able to distinguish the short wavelength cones. And in this particular pattern, we're able to identify 8 short wavelength cone terminals among the L and M cones in our volume. And once this is done, then it's possible to look underneath the cone terminals and look at the bipolar cells that are interacting with the S cone terminals. So you can imagine here, right above this region right here would be an S cone terminal. And these are now bipolar cells that are receiving direct input from the S cones. And so we're able to ultimately trace out the identities of the bipolar cells that are receiving information from the short wavelength cones. And of course, we can follow down from, these are the locations of terminals of S cones here. We can follow down and ultimately then to find the ganglion cells that are receiving input from the short wavelength cone bipolar cells. And in this case, the purple cell here is a small bistratified ganglion cell, of course, and we can verify from the circuitry that it has S versus M plus L opponency. And as a matter of fact, we can go through and we can reconstruct an entire network of small bistratified ganglion cells, all that have S versus ML opponency. Well, that's not the only ganglion cell that had been proposed to have S versus LM cone signals. There was a report that the macaque retina contains an S cone off midget pathway. That is an off bipolar cell and ganglion cell that connects directly to the S cones. So that was one report, but it was controversial because at least in the marmoset retina, there was a report that the S cones do not contribute to the off midget pathway, at least in the retina marmosets. So in order to resolve this, we wanted to go and just check to see if there were off midget bipolar cells that were connecting to the short wavelength cones in our macaque monkey retina. And indeed, we found that every single one of our S cones is associated with the output, has its outputs to an S off midget bipolar cell. And so here, each one of these blue discs just represents the terminal of one S cone, and we can see that beneath every one of those, there's an S off bipolar cell that also would be carrying S versus LM cone signals. So there you have two different ganglion cells. Oh, a couple other things. One, it's possible then to reconstruct, an entire circuit that gets information from the short wavelength cones in the retina. And here is a fuller circuit. It shows both the S on and S off bipolar cells coming from the S cones. And not only that, shown in red are the H2 horizontal cells, which are horizontal cells that are dominated by S cone signals. And finally, just for comparison, we have included a few in the gold color, a few LM off midget bipolar cells, so you can see what those look like. And also there's H1 horizontal cells that are shown in green that just carry information from the L&M cones. And if we continue to follow down from the S off bipolar cell, we find that each S off bipolar cell also connects to a midget ganglion cell. So this means that there are midget ganglion cells that has S off responses that are comparing S versus LM cones. Well, there's another report of a kind of ganglion cell that gets S versus LM cone opponent responses, and that is the M1 intrinsically photosensitive ganglion cell, or IPRGC. And so Dennis Dacey's lab had reported that in the monkey that IPRGCs, at least the M1 type, have this S off, LM on kind of response. But no one had really ever worked out the circuitry of how it is that the melanopsin ganglion cells might get to have a cone opponent response. And so we were able to, again, now since we had identified all of the S cone bipolar cells in this little patch of retina, then we're able to follow down through, and each one of these cyan forms is actually the terminal of an S cone bipolar cell. And so we're now able to follow down to the terminals and see where those S cone bipolar cells are synapsing besides on the small bistratified cell. And what we discovered is that there's a a particular kind of amacrine cell that exclusively gets all of its input, its excitatory input from S-cone bipolar cells. And so there's 4 of these amacrine cells reconstructed here. And once we reconstructed those, we can follow the output of this amacrine cell to a M1 IPRGC shown here. So now we have a circuit S cones that ultimately comes down to these terminals of the S cone bipolar cells, then the S cone amacrine cell, and the S cone amacrine cell then outputs to the M1 IPRGC, conferring S versus LM opponency. So now we see that there's another kind of ganglion cell in our retina that has S versus LM cone opponency. Well, now that we can see all the different kinds of ganglion cells that are getting input from the S cones, we discovered that there was yet another kind of ganglion cell that got exclusive excitatory input from the short wavelength bipolar cells. So we discovered a new wide field retinal ganglion cell that was receiving the very same type of cone opponent as the small input as the small bistratified cell. So shown here are 2 of these wide field cells, and we also are showing you the terminals of the S-cone bipolar cells. And this diagram down here, every one of these little blue circles shows the location of a ribbon synapse providing excitatory input directly from the blue cone bipolar cell to these particular kind of ganglion cells. So if we think of the circuitry here, the circuitry of these wide field cells, has S-on input in much the same way that there's S-on input that comes to the small bistratified cell. So this is another blue-on ganglion cell in the primate retina that we discovered in these experiments. And just for interest, that here's a side view, and just comparing the anatomy of this wide field of blue-on ganglion cell to the anatomy that's shown in red here, to the anatomy of a small bistratified cell. Well, it turns out that this newly discovered blue-on ganglion cell turned out to be the primate M2 IPRGC. So but unlike the M1 IGC that is S off, that these cells are S on. So there's 4 different kinds of ganglion cells in the retina that all have S versus M plus L opponency. Well, there's another report of a kind of another ganglion cell in the primate retina that has S versus L opponency, and that is the large bistratified ganglion cell, as reported by Dennis Stacy. And we also were able to reconstruct one of these very large ganglion cells in our retina, confirming that we also could find a 5th kind of ganglion cell in the retina that has S versus LM opponency. So, looking at the connections in the retina, we find circuitries of 5 different ganglion cells that all have some kind of S versus LM opponency. The small bistratified retinal ganglion cell, the S off midgets, the M1 IPRGs, the M2 IPRGs, and the large bistratified RGC. And I want to say that this isn't even an exhaustive list. We still are reconstructing other ganglion cells in the retina that have input from S-cone bipolar cells. So the really surprising thing is that all of these 5 different kinds of ganglion cells, because by virtue of their S versus L plus M opponency, would be strongly differentially activated by these 2 different stimuli, a 430 nanometer light compared to a 470 versus 460. Yet, this activity that has to be going on in all these ganglion cells doesn't result in a conscious percept of hue difference between these 2 things. As a matter of fact, it doesn't it doesn't result in a conscious percept of any kind. So we want to argue that these results are counter to the idea 2, that ganglion cells are primarily concerned with conscious visual perception, because here we have ganglion cells that all must be very active, when confronted with a particular set of stimuli, but ultimately don't produce any kind of a conscious percept, at least not one of hue. As a matter of fact, we would argue that actually most ganglion cells are concerned with other functions than conscious visual perception. Okay, so back to idea number 1 is that the brain has access to all the information in the retinal image. Well, vision is concerned with the pattern of activity across the retina in space and time. But we don't have conscious access to the hue information available in the pattern of cone activations that drive our many S versus LM cone opponent ganglion cells. So we all have this illusion that we have conscious access to all the information available in our retinal images. But actually, it appears that we have only access to a tiny fraction of the information that's actually available in our retinal images. And finally, idea number 3 was the efficient coding hypothesis proposed by Barlow that said that the information should be highly uncorrelated across different ganglion cells. But much to the contrary, the hue information that's encoded by all 5 types of the S versus LM RGCs is highly redundant. And yet we don't have, but none of that information is available to our consciousness. So we want to argue that we have to abandon the highly intuitive idea that the goal of our visual system is to encode all the information from our retinas and transmit it to brain regions concerned with conscious perception. In fact, the majority of the operation of the visual system is concerned with subconscious visually driven behaviors. These include visually driven motor movements and subconscious reactions to visual stimuli. So we conclude that retinal ganglion cells are feature detectors, and the S versus LM retinal ganglion cells discussed here, they project to several different places in the brain, and they serve a variety of purposes, allowing wavelength information to contribute diverse functions other than conscious hue perception. Apparently, these include ganglion cells that serve as dawn dust detectors, which can use spectral information in circadian photo, can use spectral information for circadian photo entrainment. Then could probably include motion detector ganglion cells, which use spectral cues in the guidance of orienting movements, and by edge detectors, which use spectral cues in the segmentation of objects. And with that, I'll stop and thank my funding sources, and also give credit to the postdocs and the graduate students that we've had in our lab over the years that have contributed to these ideas. And with that, I'll stop.